Who's killing the juice?
My bad, bro. Low on ATP. Note disclaimer.
If you watched that little video there, you probably were kind of confused as to what's going on. Well, that little clip was to represent the different organelles that party inside a cell. See, if you take a cell and you cut it open, what happens is you see all the different types and parts of the cell, including things called a mitochondria or a nucleus, which, don't worry, we'll dive into in just a second. But for now, what you need to know is all those different parts work together collectively to perform very specific jobs that benefit the cell overall. And the thing is, each organelle has its own functionality that it carries out. And together, they help that cell survive, grow, and carry out different life processes. So in that little clip, you saw the nucleus really reaching out to the cell saying, what is going on? In which what is called the mitochondria plugs in because it's considered the powerhouse of the cell. Don't worry if it doesn't make any sense. All you need to know for now is that there are different organelles which look very different, but they have very specific functions which collectively work together to ensure the functionality of that cell. When you're preparing to learn about the cell, make sure to understand the similarities and differences with the different organelles alongside with its cohesive structure and functionality. Please understand that these organelles work together to basically ensure that the cell functions somewhat like a factory. And the thing that you need to understand is for most biology tests or most AP biology exams, you will be expected to understand in a multiple choice format or in a free response format the similarities and differences between each organelle. This table covers that in general. Now we're going to dive into where you find it in the cell and more details as they relate to each other, but I wanted to point this out so that way you can basically review the organelle, the structure, and the function in general.
Hey, want to know why I wear jeans? Because I'm full of genes. I hold all your genetic material wed. I'm the blueprint, baby.
Before we discuss the organelles within the cell, it's important to understand the differences and similarities between eukaryotic and prokaryotic cells. So both types of cells have a cell membrane and they have cytoplasm that exist, but prokaryotic cells are considered to be less evolved. In fact, there's a theory called the endosymbiotic theory, which basically states that prokaryotic cells, which lack a nucleus, are a lot smaller, and have no membrane-bound organelles, eventually evolved to what we now call eukaryotic cells, which, by the way, have some sort of oxygen. They have a true nucleus, they're a lot larger, and then they have membrane-bound organelles. So when we're talking about these specific organelles, we're talking about this idea that we have the different organelles within the eukaryotic cell. Now, let's dive into the nucleus. So when we talk about the nucleus, we talk about this key structure right here, which is very spherical. And inside the nucleus, you'll have a little tiny spherical shape called the nucleolus. So that's this green feature here. Think of nucleus inception, where you have a nucleus within a nucleus, essentially. And basically, it has the same physical features. It's a dense spherical structure, but it has no membrane on the outside, unlike the nucleus, which has that little yellow shape structure that's supposed to be the membrane. So the nucleus is basically found in the nucleus of eukaryotic cells. It's dense, it has that spherical shape, and it doesn't have that membrane that the nucleus itself has. And the main function of the nucleolus is to control ribosome synthesis. And so when you hear the term ribosomes, I want you to remember that it's essentially created in the nucleus, but more specifically the nucleolus. And the reason why it has no membrane bound outside the nucleolus is because it has to release those ribosomes. Now, with the nucleus, you have a double membrane That's this yellow type structure here. And it acts as a control center. It regulates your growth, your metabolism, and reproduction due to the genetic material that is there. So it stores that genetic information that leads to directing cell activities. So it contains inside there RNA, which moves to the outside. Remember, in the nucleus, you have DNA. We talked about this in this process of DNA to RNA, but remember, DNA is contained and stays within the nucleus, but DNA will turn into RNA, which will then create protein at some point. That process involves RNA leaving that nucleus and traveling outside the nucleus itself. So that's important to note when you're thinking about the bigger picture and really creating a foundational understanding between Unit 1 and the understanding of DNA and RNA, and now understanding how it all fits together. This idea that we have the nucleus, and in the nucleus you do have DNA, which is the genetic material, but the nucleolus contains, or creates, I should say, ribosomes. And that nucleolus that contains ribosomes will help assist in creating RNA, converting the DNA into RNA, which then will release outside the nucleus itself. If you're planning to review this video, please note that you will be covering what's known as the endomembrane system. And the endomembrane system is a unique system that basically holds multiple organelles, including what's known as the nuclear envelope. While you'll cover the other organelles later on in this video, please understand we're covering the nuclear envelope now because we just covered the nucleus. Remember, we said that DNA turns to RNA, which then turns to protein at some point, where the DNA is housed inside the nucleus itself, and you have DNA converting into RNA, which then is released to then make protein, also known as the central dogma. Well, the nuclear envelope is the yellow part here that has various pores that allow for items such as molecules to move in and out of the nucleus itself. And it's very important in this endomembrane system because it's a boundary that basically connects the inside of the nucleus to the outside of the nucleus, allowing for materials like RNA and proteins to then release from the nucleus for further process. Let's talk about the location of ribosomes before we get started. So if you remember, here is the picture of our cell. And in the cell, you have this structure here, which is called the ER, or endoplasmic reticulum. That's this purple-like structure here. And you could have little microscopic dots on them, which are considered ribosomes. And in fact, that makes it the RER, or rough endoplasmic reticulum. We'll be covering this organelle in just a second, but I just want you to distinguish really quickly the reason why you have RER, or rough endoplasmic reticulum, versus smooth ER, or SER, which it has no ribosomes in them, is this distinguishing factor of actually ribosomes. So the rough ER is going to have ribosomes on them, and the smooth ER is not. And when it comes to ribosomes, they can be free floating outside in the cytoplasm here. That's where you see these little microscopic dots, or they can be on the endoplasmic reticulum, which is the purple-like structure, essentially making them the rough ER. So ribosomes are these really tiny structures, and they're composed of two things: RNA, or ribosomal RNA specifically, hence rRNA, and proteins. Usually when we talk about the shape of ribosomes, they usually look something like this, where they have a large subunit and a small subunit. And ribosomes are very unique. You don't just find them in the eukaryotic cell, you can find them in bacteria, archaea, and other eukaryotes as well. So ribosomes are not considered to be a lack. So they are contained in other types of cells as well, which is why ribosomes are so unique. And if you remember in the central dogma, we talked about DNA turning into RNA, which eventually turns into protein. And the thing is, when we're talking about that RNA turning into protein, we're talking about this process known as translation, which has its own dedicated unit in of itself, but it's a key step when it comes to what we call protein synthesis. So I want you to think of this idea. Remember, we talked about in Unit 1, the central dogma, and we talked about DNA turning into RNA and turning into protein. And if you remember, DNA into RNA occurs from the nucleus, and then when it turns into RNA, the ribosomes leave that nucleus. And then RNA to protein is where we have the ribosomes that essentially take place. And we have a large subunit and a small subunit, which participate in this process known as translation, which is a key factor in creating these eventual proteins. So if you look at this picture right here, which you're going to get more details in just a second, what happens is there's a specific type of RNA called tRNA, and it has the amino acids that attach to it. And if you remember amino acids are what makes up what? Do you remember which macromolecule? If you said proteins, you are correct. And remember, you have amino acids, which are the building block of proteins, right? And so what happens is they have to create a chain, and then the ribosomes help them to create this chain by taking the RNA code and then eventually turning into protein. I also want to preface this by saying that translation is a lot far more complicated of a process, but like I said, there is a whole unit dedicated to this in total. Because of this, I'm not going to focus in on the process of translation. For now, you just need to know that in the central dogma, you are going from RNA to protein. Ribosomes help assist this process with their large subunit and small subunit to ensure that the RNA eventually turns into protein. So that is where you want to know for right now what ribosomes do. They're common in every living cell, and it's suggested that they are a common ancestor for all life on Earth, which is why they're considered to be in all of these different structures. This is considered a reason why we believe the theory to be true. And remains to be unchanged. There's not much change in the structure. We have the typical large subunit and small subunit, which is what makes it very unique in terms of its shape, that it hasn't really changed over a period of time. Now, ribosomes are a little bit complicated. There are specific types of reasoning where ribosomes come into place as well.
Translation complete. Protein ready for action.
Let's tie this all together because in AP biology, you want to remember that all the units connect together. If you are eventually going to get some sort of FRQ or multiple choice question, you will be expected to understand not only Unit 2 in terms of just organelles, but how it relates conceptually to earlier and later units. So let's try to tackle that with ribosomes. So if you remember, we talked about DNA going to RNA. And DNA to RNA, remember, this occurs in the nucleus, and mRNA is going to basically go from the nucleus to what's called the cytosol. So if we have a cell, and remember the nucleus, we have our DNA here, and it's going to transfer to the RNA, and then the RNA is eventually going to go out. And how this is done is through the central dogma, which undergoes three main steps: transcription, translation, and then finally protein synthesis. Please note there is a whole dedicated unit just to this process, and we will cover this more in detail in a later time. but I want you to remember the understanding of where ribosomes really come together. So when it comes to rRNA or ribosomal RNA, essentially it's creating ribosomes, and they're going to have like the large subunit and the small subunit together. And what happens is that is where you're going to see where the messenger RNA is going to come in. So what I mean by that is even though we have rRNA, mRNA, we have mRNA that is a different type of RNA, which basically connects the genetic material. So we have mRNA, and then we have protein synthesis, which allows us to take our ribosomes and create the genetic code, which leads to what's called translation. So to recap, we have DNA, which then goes to mRNA or messenger RNA. So that's this right here. And what it does is it takes the DNA code and transcribes into what's called messenger RNA. And if you remember in Unit 1, I told you that DNA is double-stranded while RNA is single-stranded. Well, that's because you sometimes have a DNA code which will then create an RNA code, which is essentially what is happening within the nucleus. So remember, I did say in Unit 1 that you have the DNA in the nucleus, and it starts off that the RNA is going to be there. That RNA that we're talking about is the mRNA. The process of taking the double-stranded DNA and taking one of the sets of nucleotides, so A, T, C, G, and combining it to its complementary RNA strand. So A will attach to U, because remember, we don't have thymine in RNA. And then thymine will connect to A, and then C will connect to G, and G will connect to C. So this is the process of going from DNA to RNA, specifically messenger RNA, and this is all occurring inside the nucleus. Okay, so that's how we tie in Unit 1 into now. Now we have the mRNA. So right now we're inside our nucleus. The mRNA has to go to the ribosomes. So basically it is going to travel externally, go to those free-floating ribosomes, right? Which is composed of, remember, rRNA. So that's this. And it's going to have instructions that are now interrupted. So the instructions are going to basically say, all right, we have our RNA code. You now need to form proteins. And that's where protein synthesis comes in. You have these amino acids. Remember, we talked about those different types of amino acids. For example, you could have an amino acid that says start or AUG. And basically, it will provide a story to eventually create and link the amino acids to eventually create, if you remember in Unit 1, a polypeptide chain. So if you can see now, we talked about amino acids and we talked about how they're the building block of proteins. And the goal is to create proteins. And the step of taking those amino acids, which is our building block, and creating the polypeptide chain is done through translation. So that's just a little recap for you before you cover it again later in later units, but also tying it into Unit 1, where you had a whole section dedicated to proteins. So this is how it relates together in terms of steps within the cell itself. When we're talking about plants versus animal cells, I've noticed when we're reviewing this unit as it's been updated with AP biology or college biology, there really isn't a strong focus on plant cells. And I think the reason why is because there's a whole unit dedicated to plant cells. And while that was the case before, where you learned about things like respiration versus photosynthesis, which you'll cover in a later unit, I think the focus is trying to compare and contrast plant cells versus animal cells in terms of organelles and understanding on a bigger scale their importance. So what I mean by that, yes, you will need to know that the cell wall is relevant for plant cells. They're necessary, and they are an organelle that is in plant cells that you don't see in animal cells. And the reason why that is because it has to uphold a very rigid shape. The plant cell has to hold that up rigid shape to ensure that the plant can be able to essentially be stabilized. And so when we talk about cell wall, I want you to remember that animal cells do not have a cell wall, and that's because they have to be more fluid, they have to be more irregular. And so the relationship is that cell wall and that rigid shape causes it to have also a rectangular shape. Versus with animal cells, you don't have a cell wall. And so cells can be very irregular in shape and more easy to move around. There's also a structure in plant cells known as chloroplasts, as you can see in the table here. And chloroplasts have this green structure. And the reason why they have that green structure is because it gives that green color to plants, but that's because it's trying to harness the sun's energy, which you're going to cover later on in a future unit. They also have vacuoles that exist both in plant and animal cells, but it's larger in plant cells. And you want to think, why would that be? That's because it has to uphold or hold in a lot of water. We can easily, as animals, go ahead and take water on a repetitive basis, but plants don't have that luxury, and therefore they're going to need a large central vacuole to uphold that water, especially during times of drought. So we also talked about the shape and the relationship between that and the cell wall, but I also want to talk about centrioles, which is a very specific structure that you're going to see in the process of cell division when it comes to animal cells. Plant cells have a very similar process when it comes to replicating cells. However, they are rigid in structure, so they have to divide a little differently. They don't use something called centrioles. So once again, you can see in this table the key similarities and differences between plants and animal cells, more specifically the differences, and how those differences allow plant cells to have very specific functions and animal cells to have their own specific functions as well.
Fam, I'm out here running marathons making an ATP, and y'all still complain you're tired. Bro, you are the powerhouse. Stop crying and keep cranking out that energy. The muscles outside are wielding.
Some might argue that the nucleus is one of the most important organelles, but in fact, some others might argue it's the mitochondria. See, the thing is with the mitochondria, we have a whole theory dedicated to why it even exists and why it's so complex of a structure. And that's known as the endosymbiotic theory. Now, you probably heard this in Unit 1, and you'll probably learn this in a later unit, specifically one dedicated to cellular respiration, and you might even hear it in the evolution-related units. Well, let's recap. So with the endosymbiotic theory, there was a theory that once upon a time there were simple prokaryotes, and those simple prokaryotes did not need oxygen to sustain itself. And what had happened was over time there was a mutation where a complex organelle known as the mitochondria had evolved, and a prokaryote had engulfed this mitochondria that allowed it to become what we now know as eukaryotic cells. So the mitochondria is very unique. It has its own DNA. If you remember in Unit 1, DNA and RNA allow for genetics. Well, think about that. The mitochondria has its own DNA. It has its own genetics, which is crazy. And it's also very complex of a structure. In fact, it has its own direction as to how it reacts to things within the cell. So when we talk about the mitochondria, I want you to remember the term ATP. If you've taken honors biology or any sort of biology, you probably heard of this term. And I want you to think of it as energy that is produced within the cell. There is a process known as cellular respiration. I want you to think of breathing. So when you breathe, what happens? You breathe in and you breathe in oxygen and you breathe out CO2. Well, that process is highly involved in the organelle mitochondria. In fact, there's a whole unit dedicated to cellular respiration, and so you'll cover these steps listed here even further. So there is a process known as glycolysis, which is the first step in cellular respiration, and this occurs outside of the mitochondria. in that outside process, you have pyruvate, which is produced, and it enters the mitochondria and then starts a process known as the Krebs cycle. And this occurs in this structure called the matrix. So the organelle mitochondria has its own unique structures, such as the matrix. And the matrix is this little opening here, and it's like kind of like it thinks of similar to a maze. So if you think of a maze, when you go across a maze, there's these openings that allow you to walk through. If you go through like a cornfield in the fall, you might have seen one. I want you to think of that when you're thinking of the matrix. So what's happening here is you have the Krebs cycle that's going to happen in this panel of opening. And that starts one of the processes. So after the Krebs cycle is complete, what happens is you have what's called the electron transfer chain. The electron transfer chain is very important because it produces a high amount of ATP or energy. And this occurs in what's called the inner membrane. So you have the matrix, and then you have this inner membrane here. And then what ends up happening is in that inner membrane, afterwards, the ATP synthase, which is a enzyme, the way you would remember that is, remember in Unit 1, we talked about ASE endings and understanding that that is a clue indicator to let you know that that's an enzyme. Well, it uses what's called a proton gradient to basically generate that large amount of ATP. So in the electron transfer chain, that final process, which occurs in that inner membrane, you are generating that high amount of ATP. Now, let's talk about the other structures here within the mitochondria. We talked about the matrix and we talked about the inner membrane. What about the outer membrane, which is right here? So that outer membrane allows the mitochondria to be a double membrane. It's a very smooth surface, unlike the inner membrane, which has all these foldings in here. And the outer membrane is there to provide an extra barrier, and it regulates basically what enters and exits the mitochondria. Now, you might think to yourself, the mitochondria acts like the cell itself, and it does. It's actually very interesting that it has that double membrane, and it regulates what enters and exits the mitochondria. And with the inner membrane, it is highly folded, unlike the outer membrane, which is smooth. And in that inner membrane here, remember, you have ATP production. So to compare the difference between outer and inner membrane, outer membrane is smooth, allows for a protective barrier, and it regulates what comes in and out. And then when you're talking about that inner membrane here, we're talking about it being very folded, and the folding is important for the process of the electron transfer chain, which then produces ATP with the help of the enzyme known as ATP synthase. Now, you have the matrix, which we said is inside there. Remember, it is fluid filled. Okay, so while it's inside that inner membrane, it's going to be filled with fluid, and it's important for the Krebs cycle. Now, let's talk about this cristae over here. So this is the term cristae. When we're actually talking about cristae, we're talking about these I want to say you're looking at the folds. So this is a fold that's there. So more folds are important. So this is a fold. This is a fold. So that's what we're pointing to, the different folds that are here. And the folds are important because they allow for not only greater surface area. So if you want to think of surface area, you want to think in terms of this idea that you have more space essentially, and it allows for more electron transfer chain proteins and ATP synthase enzymes to increase the production of ATP. So all of these folds here, not just this fold, but this one here that it's pointing to, or this one and this one, is really important because it allows for more openings for ATP to be produced, which is great because that's the purpose of mitochondria, to produce the most ATPs.
I know you're always busy making proteins, but tonight, can you just focus on me?
Only if you promise to keep things smooth.
Let's talk about this system known as the endomembrane system. The endomembrane system, if you could see the root word here, endo or enter, you can think of enter, and you could see endomembrane. So basically what's happening here is you have a group of various organelles, nuclear envelope, endoplasmic reticulum, Golgi apparatus, vesicles, lysosomes, and the cell membrane that cohesively work together to ensure that whatever comes inside the cell or exits the cell is doing so in a safe way. Now, we are going to talk about the specifics of each of the organelles throughout this video, but I wanted to cover what they look like physically before we cover the functionality. So, for example, we covered this before. This is the nucleus, and the nucleus, remember, has the DNA. And DNA has to exit through RNA, right? And we do that through the nuclear envelope. And so outside the nucleus is this membrane known as the nuclear envelope. And we have something called the endoplasmic reticulum. Now, if it's embedded with what we have known as ribosomes, remember we talked about ribosomes, that is considered the rough ER. If it doesn't have the ribosomes attached to it, we call that the smooth ER. We also have these little bubble-like structures. Those are your vesicles and lysosomes. And then we also have the Golgi apparatus. Now, all of these different membrane-bound organelles work cohesively together to ensure that products can enter and exit the cell in a cohesive manner. Now, I want you to remember Unit 1, where you talked about macromolecules and understanding that these are the things that are coming in and exiting the cell, so in and out of the cell, basically. And those include, remember, macromolecule proteins and lipids. And if you remember, polysaccharides are covered under what we call carbohydrates. So they're all entering and exiting the cell. But we have to ensure that the safety of the cell exists. And to do so, we have to ensure that we're entering or exiting various items in a safe manner. And that's what these different organelles work together to do. So first, let's talk about the endoplasmic reticulum. Now, the endoplasmic reticulum is part of that endomembrane system. And if you remember, we could either have smooth ER or rough ER. The rough ER is embedded with what we call ribosomes. And they're a complex network of systems that is located within the cell. So this is a picture of the cell. And remember, this is the nucleus. And you can see here, you have the endoplasmic reticulum really close to that nucleus. And if you remember, what leaves from DNA, or sorry, from the nucleus itself is not going to be DNA, is going to have DNA, which turns into RNA, which is immediately going to leave. And if you remember, RNA is associated with ribosomes, right? So the endoplasmic reticulum, it makes sense that it's so close to the nucleus because it needs to work cohesively with the nucleus to ensure that it's working to make sure that the materials such as proteins and lipids are leaving or entering the cell in a proper fashion. Now, once again, if we have our endoplasmic reticulum, we can have smooth and rough. There are spaces in between, so it's like a sandwich with opening in it. I want you to remember that's the cisternal space. I never can pronounce that correctly, but you should understand that that's a space or an opening that is within it, sort of like a scrunched up belt or scrunched up sandwich where it has a opening or space of air, but not air, but has different items inside, but it is an opening. And if it's rough, it has the ribosomes, which makes sense because remember, we're producing those ribosomes near the nucleus's function. So that makes sense why the endoplasmic reticulum is so close. And then we have the smooth ER, which basically is going to be no ribosomes attached to it, right? It's very smooth, hence the reason why it's considered the smooth ER. Now, if you are given a free response question or if you're given some sort of multiple choice question, you oftentimes are given the answer choice of rough versus smooth, meaning if it's a multiple choice question, they're going to ask you to distinguish somehow the difference between rough and smooth. And students often forget that they have to compare between the two. So I created this table to help you understand the similarities and differences. So for example, if we're taking a look at the rough ER, it's going to look rough because it has those spots, like it has those like the white spots right here, they indicate that they have ribosomes, and they look rough when we look under the microscope. Now, with the smooth ER, remember they're smooth because there is no ribosomes. Now, let's talk about the functionality between rough and smooth because this can be difficult for students to understand, especially if you're given a free response question or multiple choice question. So, when you're taking a look at a cell and you have the nucleus, it usually faces near the rough ER first, and that's because remember, when we think of nucleus, I want you to think central dogma, DNA to RNA to protein. So it makes sense why the rough ER is so close to the nucleus because it's required or necessary for protein synthesis. And that's how you're going to remember rough ER. If I have the rough ER, it's closest to the nucleus. It's rough, so it needs a little help. That's a way you can remember it. And it needs help, so it needs to be close to its bestie, the nucleus, and that will allow it to basically produce protein through protein synthesis. Smooth ER, I want you to think of having as lipid synthesis. Now, if you remember, we talked about fats and steroids. I want you to think about when we were talking about macromolecules and specifically that section of lipid synthesis. When you're thinking of lipids, you're thinking of oils or you're thinking of things that are smooth, and that helps you remember smooth ER. You can think about those steroids and how gym individuals will bulk up with steroids. And so the way that I remember it is thinking that, okay, if I bulk up, I'm going to try to be smooth for the ladies, essentially. And that's going to be your lipid synthesis. It's not a great way to remember it, but it helps you distinguish the differences between rough and smooth ER. Other things to note is that the rough ER, because of its major breakdown, it's going to work to send the proteins through what are called vesicles to eventually lead to what's called the Golgi apparatus, which we're going to talk about what specifically the Golgi does, but you want to remember that the rough ER is rough because it's creating protein and it's going to eventually go to the Golgi apparatus to basically further continue on with the process. Now, with the smooth ER, it wants to stay healthy. It wants to be a gym rat, right? So it wants to detoxify as much as possible. So the smooth ER I always think about as detoxifying those harmful chemicals such as drugs or alcohol are going to go through the smooth ER. If we're talking about transport role, we're going to talk about the idea that they both are part of the endomembrane system and they work together. But one is for protein, specifically the rough ER, and when we think of the smooth ER, you want to think of lipid transport. The idea you want to think about when you're reviewing all of these different organelles is that they're a giant factory, and the factory has little individual factories. So in this case, the rough ER is its own factory that produces protein, and the smooth ER has its own factory that is going to produce lipids. So hopefully that helps you distinguish the difference between rough and smooth ER, especially if you get a multiple choice questions, because more than likely they're going to give you A, B, C, or D as an option comparing and contrasting rough and smooth ER. Hopefully the analogy of thinking of smooth ER as being a smooth, slick individual versus rough ER being kind of like this individual that needs to have more protein can help you distinguish the differences between the two.
Yo, Golgi! I'm fully folded and dripped out. Ship me to the phospholipid bilayer ASAP.
Let's talk about the endomembrane system and the understanding of what it could be, which includes the Golgi complex or apparatus. The Golgi apparatus or Golgi complex is very unique in the process of what it does. What it does is it basically is found in eukaryotic cells, often is stacked with what's called the cisternae, and these are flattened stacks. So you can see that they're rather flat over here, and oftentimes when you're looking at a cell structure itself, you might get confused with the endoplasmic reticulum. And the way that you can think of it is, first of all, there are no ribosomes that are embedded on the outside. Oftentimes you'll see vesicles or their circle-like structures nearby. And that's one of the key ways that you can distinguish them. Those circle-like structures are known as vesicles. The other thing is you'll see them mostly on this nearby outer membrane. So that's a way that you could tell what the Golgi complex or Golgi apparatus is. The function of the Golgi complex is somewhat like a post office. You want to think of it like a post office in which there are things that need to be shipped in or shipped out. So when you have the Golgi complex, they're going to receive from the rough endoplasmic reticulum what are essentially going to be various proteins. So they might receive proteins that follow through in the Golgi apparatus from the ER. And from there, let's say from the ER, you have a protein that enters the post office. From there, it's going to package, and it's going to basically package those proteins in a way that they can then leave and go to the correct destination. So sometimes you'll need proteins that will stay within the cell itself. Other times they have to leave the cell and go to another cell, or they need to go to another part of the organism in totality. Because of that, we have considered that one of the key functions to not only create protein and modify it and fold them to double check before they leave the Golgi apparatus, but also because they're ready to package and transport. So sometimes they'll leave outside the cell in the form of secretion, and they'll be tagged with where they're supposed to go. The golden way to think about it is, like I said, the post office, where it's just going to be processing, labeling, shipping proteins and lipids, and it's essential for cell functions. Now, if we go into detail of what the cell structure looks like we said, we have these sister rays that are there, which are these inner foldings, and you could see these vesicles here. And what happens is when you have an incoming vesicle, so incoming vesicle meaning that you are going from some part of the cell to the Golgi apparatus. So I want to preface this by saying when we're saying cis face, if you see the Golgi apparatus here, cis would be facing the nucleus. And if you have transface, that's going to be facing away, exiting. So this is going to be considered the transface. And what happens in cis face is you have an incoming vesicle. So imagine somewhere in there you have an incoming transport vesicle. And then what ends up happening is that it gets fixed in the sister ray. And then what ends up happening is you have newly formed vesicles that then have to pinch off through the transide and then eventually secretes outside the cell. So that is the unique process of the Golgi apparatus more specifically. So once again, to recap, you have the cis face that is facing the nucleus and the trans face, which is facing the outside of the cell. And when an incoming vesicle comes in from the nucleus into the Golgi apparatus, it's going to have a vesicle attaches to the Golgi apparatus from that vesicle, and then it will eventually go into the cisternae or those foldings that we said, and then comes out through the transide as a vesicle to then exit the cell. So this is what the Golgi complex or apparatus does.
Hey, I'm Lysosome, the cell's cleanup crew. I break down junk, recycle parts.
Let's talk about two key features of the endomembrane system. The first is a lysosome, and the second one is transport vesicles. With these two structures, they look very similar. So if you compare this structure to this structure, oftentimes it could be very confusing to distinguish the similarities and differences, especially if you're given a picture of a cell. And you can see it's very hard, especially if you're given a tiny picture. So we're going to try to help distinguish the differences. Firstly, if you are given some sort of exam question related to either one of these, please note oftentimes they will give you the functionality to help you distinguish the difference. So first, let's talk about lysosomes. Lysosome, and you can see the root here, word here, lyso. So I want you to think of Lysol wipes. So Lysol wipes. And when you think of Lysol wipes, if you take the wipe, the purpose is to take maybe like a desk or some sort of surface and clean it off. So next time you're in the grocery store and you are walking around, look for Lysol and think of lysosomes. Their main goal is basically to clean. So they work as a recycling truck where products will go into the lysosome and it will be captured to basically distinguish whether or not we need to decompose it as waste, damaged organelles if they need to be decomposed even further, macromolecules that have the help of enzymes to really distinguish whether or not we can reuse them or we need to get rid of them. So that's what the recycling system is very similar to lysosomes. So they will take a look at various features that come into the lysosomes and say, hey, do we have to keep this within the cell or do we have to let it go? In which we have another organelle called the Golgi, which will distinguish whether or not it needs to be packaged or shipped outside the cell if it needs to be let go of outside the cell. Now, if you have the other organelle, which is transport vesicles, which are also part of the endomembrane system, there are small vesicles which look like tiny bubbles that transport materials that help to either deliver or exit to the plasma membrane. So what I mean by that is there's something called exocytosis versus endocytosis. Now you will talk about this further in a section dedicated to the plasma membrane, but for now you need to understand that the transport vesicles sort of have this pinch-like structure that allows the cell to exit material or enter material into the cell itself. Now, when we're talking about these transport vesicles, I want you to view the left side versus the right side here. On the left side, you have an example of endocytosis. This is a cellular process that basically involves the movement of material into the cell. So it is entering the cell. So as you can see here, what's happening is there's these particles. It forms a circle-like structure. And then with the phospholipid bilayer, if you remember in Unit 1, is a part of lipids. And it works to encapsulate or form this circular structure around the particles and allows it to come from outside the cell to inside the cell, essentially engulfing, engulfing, or entering, entering within the cell. So that's how you can remember endocytosis. Now let's take a look at exocytosis, which is the opposite. They form a vesicle that probably had come from the Golgi apparatus and was told, hey, we need to exit the cell. So it is transported to the same plasma membrane, and then it sort of attaches to the plasma membrane and they require some sort of energy. So this is where the mitochondria would come in to provide that energy. Both endo and exocytosis, by the way, require some sort of ATP or energy. And they work to essentially exit these particles outside the cell, hence exocytosis. And as stated before, you will have a whole section dedicated to these processes, so I won't focus too much into it, but I just wanted to provide this visual diagram so you can see and compare the similarities and differences between endocytosis and exocytosis, which is a part of transport vesicles. Let's talk about the structure of lysosomes. In this diagram listed here, you see that they're membrane-bound organelles. And inside lysosomes, we have these little white spots here, and they're supposed to represent hydrolytic enzyme. Remember, when we're talking about lysosomes, you want to think of the root lyso, like Lysol, and that'll help you remember the main job, which includes digesting, recycling, defending, and triggering what we call apoptosis. Now, this is commonly found in animal cells, rarely in plant cells. Now, the purpose of lysosome is to break down things like food particles or organelles or cellular debris. And the way that you can remember less than 7 pH is helping you also to remember that it's very acidic, right? And our stomach is very acidic. So it makes sense that a very acidic environment is going to help you break down those food particles. Also, it participates in a process called autophagy, which means that you recycle damaged cell parts. So, say for example, there is a Golgi apparatus that's broken, it will come inside the cell, and autophagy will allow us to break it down so it creates other organelles that are useful as well. Now, we also have the term apoptosis, which is one of the main functions of lysosomes is to process or trigger apoptosis. And what that is programmed cell death. And you might be thinking, if I think of cell death, I think of something bad. Well, see, the thing is, our body has a ton of cells that continue to grow indefinitely until we tell it to stop. And the reason we don't want cells to grow indefinitely is because it will produce things like cancers, or it can hurt or hinder by producing or not allowing for the production of new cells due to the crowding of the old cells. So apoptosis is the allowing of programmed cell death to release these digestive enzymes. Oftentimes, cancer patients, for example, lack severe skills in their cells to produce apoptosis. So that's a way that you can remember. And remember, one of the other functionality of lysosomes is defense. They destroy pathogens like bacteria. So they are especially in white blood cells. You want to think immunity, and lysosomes will help basically act as an immunity assistance by also destroying pathogens that are not required or needed for the cell or organism in total. Let's talk about the structure known as vacuoles. So vacuoles are important because they're relevant for plants mostly. Often signs of a large vacuole indicate a plant cell. And as you can see, this is a sample of plant cell, and you can see how tightly wound the plant cell is as compared to the animal cell. And you can see that giant vacuole there. So with plant cells, you have a large central vacuole, whereas with animal cells, you'll have small, little multiple vacuoles, I should say. And the reason why is we have to take a look at plants and see how they garnish their ATP or energy. So first of all, they harness the sun's energy, but they also need to take into account the water and the nutrients from the soil. And in this case, what happens is if you've ever seen a plant that has been overwatered, over time, what happens is it will wilt. And vacuoles help to prevent that because they store the water, nutrients, and waste while ensuring to maintain what we call turgor pressure or rigidity, which allows a plant to stand up. You want to think about when there's a rainstorm of some sort in the wilderness, if you see trees, they're not really wilting or falling apart that easily, and that's due to turgor pressure caused by the vacuoles themselves. And so essentially what happens is you're providing the structural support. Whereas with animal cells, you're just using it as a backup storage for those nutrients, waste, and cellular processes. So let's talk about the difference between the vacuoles and what we call centrioles. So centrioles are a little bit different. Centrioles are known to have 9 microtubule tubulets, and the reason I should say triplets. And the reason why these are important is because compared to the vacuole, centrioles are found mostly on animal cells rather than mostly in plant cells. And that's due to the cellular division that occurs with a process called mitosis or meiosis, which takes into account chromosomal separation, which are these things inside the nucleus. They do lay outside the nucleus itself, but you will talk about centrioles more in a later unit as it relates to the process of mitosis and meiosis. But for now, you just need to know that they're outside the nucleus, but they help assist the nucleus when it comes to organizing those chromosomes. And they'll utilize something called the spindle fiber, and this helps forms the cilia and flagella as well. So one, they organize the spindle fibers, and two, they formulate what's called cilia and flagella. And what cilia and flagella are is if you ever look at a cell and it has a tail, or if it has a ton of little hairy structures on the outside, that's known as cilia. The tail is known as flagella. So the centrioles help form those, and they're a big structure component when it comes to that. But for centrioles, in terms of process of cellular division, they remain somewhere near the nucleus, but outside the nucleus to help assist with that cellular division. And so that's why they're that important. Now, if we take a look at the endomembrane system again, I want to focus on the plasma membrane. There is a whole unit dedicated to the plasma membrane. I want to say section, not unit, but a whole section dedicated to the plasma membrane. And we're going to talk about the specifics of that when we get there. But for now, you just need to know you have the phospholipid bilayer that remains on the outside of the cell. Sometimes they'll have these openings, and the plasma membrane basically will allow for entering and exiting of the cell different substances and is a barrier for initial allowing for things to come in and outside the cell.
Meet the chloroplast, the plant cell solar panel, engine, all rolled into one.
Let's talk about the chloroplasts. So when we think about the chloroplasts, we're found in plants and photosynthetic algae. So what we mean by that is when we're speaking of chloroplasts, we understand that chloroplasts are found in plants and they're the site of photosynthesis. And when we're talking about plants, we're talking about this idea that we have carbon dioxide and water that we utilize to basically adjust for our plants and their nutrients. And we have light that comes into play. All of these together cohesively produce glucose and oxygen, and chloroplasts help assist that process. They have a double membrane that exists. The outer is for protection. The inner encloses what's called the stroma. So the stroma is this like light green space that you see here. And the fluid inside this membrane is where a process known as the Calvin cycle occurs. So this is basically where glucose is formed. And if you remember, glucose is a type of sugar. You could tell by the ending of OSE. So it's under the macromolecule carbohydrates. And this is how we tie in Unit 1 into Unit 2. So when it comes into notifications that you're seeing something related to chloroplasts for AP biology, I want you to remember that the stroma works to produce this glucose, which is a simple sugar. This is why vegetables and fruits that you consume actually have a lot of nutrition when it comes to glucose. So we want to note that the process known as the Calvin cycle occurs here. While you're not covering photosynthesis in this unit, please note you will cover it in tandem with respiration in a later time. But for now, this is just a brief overview. So you will cover the structure of chloroplasts again when you get to that unit as well. Now let's talk about thylakoids. So thylakoids are these disc-like shapes. And the reason why they're disc-like shapes is so that way when the light hits it, it will hit it in a fashion or manner that can basically allow for the sun rays to hit a large part of the leaf that contains the chloroplast when it comes to plants. And they stack up into what we call grania. which contains essentially what we call chlorophyll. And this is where light reactions occur. So this is the first step where that sun ray is going to hit that thylakoid and convert that into a usable form, aka ATP is going to be coming from that light. And if you remember what ATP means, we're talking about energy here. So the green pigment, by the way, is due to that chlorophyll. That's why when you see in the fall and the winter, um, fall transitioning into winter, I want to say, you'll see different colors, like you'll see sometimes yellow leaves if you live in a deciduous area. Sometimes you'll see red leaves, and that's because of the chlorophyll. It's because the green pigment has to absorb the sunlight and in the process of photosynthesis. And the key reaction here is understanding that CO2 and H2O and light work together to cohesively produce that glucose and that oxygen. And so that is the purpose of what chloroplasts are. Make sure to pat yourself on the back because you've made it thus far. This is probably one of the giant videos that I have to create as it relates to all the units combined. And that's because these key organelles are not just important for this unit, but units later on. Take, for example, processes like cellular respiration depend on the mitochondria, while the chloroplast depends on processes like photosynthesis. You'll learn about in later units cell-to-cell communication, so understanding the plasma membrane and its functionality is important as well. Also, great job in understanding the various differences when it comes to the endoplasmic reticulum and their processes, because that will come up in later videos as well. Before we end, I wanted to provide this summary table of what we had covered. We're also going to cover a key question, but before we get started, make sure to review this and cover the table. As always, you can review the video at a higher speed, but make sure to understand the key concepts of their organelles and how they relate to one another and their location within the cell. They'll help you really tackle those free response questions or really difficult multiple choice questions as well.
Thank you for learning about Section 2.1.
We hope you had fun touring our cell factory.
Let's try to see if we could tackle the following question. Take a moment, pause your video, and take an attempt to answer the following question. Okay, now that you're done reviewing the question, let's see if you came up with the correct answer. So did you come up with answer choice B? If so, you got the correct answer. We know that the mitochondria does help generate ATP. Ribosomes do help synthesize proteins. But let's talk about perioxysomes, which do help break down fatty acids and detoxify harmful substances. We did not talk about this as an organelle because while you're required to know this, it isn't something that is really a big topic, but you should be familiar with it. Now, perioxysomes are known for various metabolic reactions, particularly in lipids and hydrogen peroxides. And if you remember, we talked about lipids and we talked about fatty acids. And remember that for fatty acids, we have to synthesize lipids. And this is important for neutralizing what we call hydrogen peroxide, which is considered very toxic. So this makes sense why we say detoxify harmful substances with perioxysome. But it's talking in the question about a biologist who's examining something that digests. So digestion is lysosome. If you remember Lysol wipes, and it says digest damaged organelles, and it's going to basically membrane bound, has hydrolytic enzymes, and functions best in acidic conditions. That's your key word here, acidic conditions. And if we remember, lysosomes are known for digestion, and they're coming from the Golgi apparatus. So they have to work to basically break down cellular waste. So this would be the best suited answer. I just wanted to throw this question in here in case you get a question where there's an organelle that you did not review, in this case, perioxysomes. So the best answer choice is one that talks about digesting damaged organelles, membrane-bound, hydrolytic enzymes. Acidic conditions is your key indicator to indicate that the best answer choice is answer choice B or lysosomes.
Appreciate you riding along for our cosmic journey. If today's mission brought you the knowledge that slaps, make sure to hit that like button to boost that signal. Subscribe if you're locked in for our next launch. And don't miss the link tree. It's got all the goods for AP Bio, PSAT Math, and SAT Math. Before I sign out, make sure you're all in one orbit and diving in. Killing out. Till next time.